Now, you all already have the summary slides, but I'll be adding more things, okay? So again, I also gave you your notes. So I'm going to talk about this immunity. Immunity always come up with three types, okay, of immunity questions. So first type of immunity questions, they always like to test, okay, what is the difference between the innate and your adaptive, and you're supposed to elaborate on both innate and adaptive immunity, okay? Second thing, we also need to take note under your adaptive, okay? Then we also need to take note about your antibodies. You are expected in syllabus to know what's the structure of antibodies and the functions of antibodies and how they actually have different variety of variations, all right? And then we always have your infectious disease. Later we'll talk about that, as well as your vaccinations and antibiotics, okay? So, again, without further ado, first thing let's talk about is your innate, okay, immune response. For your innate immune response, I already mentioned, okay, innate is always like your first primary immune response, is your first line of defense. Please take note, for innate, we always have two types. First is actually your external barrier defense, but I'm going to elaborate on this because I've seen some schools, a couple of schools actually, which actually talk about what is your external barrier defense. All right. Sorry, let's see. It comprises of, again, three types of your cells. First one is obviously your epithelium layer. Whenever we want to talk about epithelium layer, this is the one that provides the impermeable, okay, physical barrier. Alright, so this impermeable physical barrier prevents entry of your pathogens like virus, okay, through cell-to-cell barriers. So this is the first one. Alright, sit there. So second thing, let's take note. We have here, your second one is your mechanical defense. For mechanical defense, right, is your combination by your secretory cells. You have secretory cells or gland cells, okay, that is always found sometimes in your airways of epithelium, all right? Idea is that they have combination of two things. Number one, they actually have combination of mucus production, all right? The idea for mucus production is to trap pathogens and prevent them from entering the cells. The other one is your cilia. So I repeat, first one is your mucus production that you learn in O levels actually. And the other one is presence of cilia. You all should know already, cilia will help to move the mucus, all right, of your trapped pathogens out of your respiratory tract. Idea is that you want this to be killed by your acidic conditions of your stomach. So take note, your stomach contains HCl, that's why we'll kill all these pathogens here. Second one, your epithelium cells able to secrete this called your antimicrobial defensin. Antimicrobial defensin is actually a substance, alright, that can prevent, alright, your pathogens like viruses from entering the target cells. So they are just anti-chemical substances, alright. And then lastly, sometimes you can say how your epithelia also can provide your mechanical barrier because you can expel your pathogens via cilia or induce coughing. So that's why we always have coughing. So this is the first one, your external barrier defense. Always think about these three things, all right? If you want to elaborate, these are things you can look out for. But we want to focus on is your internal defense. So I said already, internal defense, okay? For those who are with me before, I already mentioned they are always got to do with these three things, your inflammatory response, okay, your phagocytosis and your natural killer cells. But I say phagocytosis is the most important one, right? Agree? Please take note for your internal defense, what are the cells that are usually involved. Cells that are usually involved obviously is your phagocytes like macrophages because they need to perform phagocytosis. Also includes dendritic cells or other immune cells. We also have your natural killer cells. Okay, natural killer cells are the only lymphocytes that are able to induce apoptosis, and they are the only lymphocytes part of your internal or innate response. All right. We also have, again, macrophages. I purposely talk about macrophages even though they are part of your phagocytes, because later you'll see macrophages not only can be your antigen presenting cells, but they also secrete this thing called pyrogen. Pyrogen actually is secreted by activated macrophages. Later I'll talk about that. Is to rise or increase the systemic body temperature. So let's talk about the first one. Your internal defense, first thing, so I already mentioned, is your, hopefully you know, is your cytokines and chemokines that is secreted by your this macrophages. Alright, so we call that actually your cytokine or chemokine secretion. So let me just write first, this is under inflammatory response. Idea, right? I repeat again. We have the first one called your cytokine and your chemokine secretion. Okay, so again, let me just elaborate. So you just tell yourself this one is released by your macrophages. So macrophages, okay, that actually secrete cytokine and chemokines. Please take note, whenever I use all these terms, they are just very broad terms. It's like hormones. Hormones is a broad term, but there are many different types of hormones, okay? So, idea, what's the purpose of these cytokines and chemokines? First one, I'll just elaborate. First one, whenever we want to secrete, is to increase permeability of your blood vessels. Especially your blood capillaries, alright, which will actually result in inflammation. This is the one that results in what we call inflammation. That's why sometimes we have sore throat or headache here, alright. But the second one is your macrophages. Besides having this, they also, under your inflammatory response, activated macrophages actually secrete pyrogens. Okay. Now, what's the purpose of pyrogens? Same thing, is to lead to rise in your systemic body temperature. Usually that's why you have fever. The reason why they need to secrete or increase your systemic body temperature is because if your body temperature is faster, okay, you know, enzymes, everything have higher kinetic energy, you have higher, okay, reaction taking place, and this is what we want. We want a response. Body temperature. Blood. Okay, so this is the first one. Clear? Now, besides this, do take note. So we have eventually your complement activation, because that is actually to bring about, I said the second one, very important, is your phagocytosis. I don't have space, I'll just write here, but they are not part, it's part of innate. So phagocytosis, I said already. More often at times, they always like to test about phagocytosis because it's also involved in things like your tuberculosis, mycobacterial tuberculosis. And I say, for phagocytosis, if you ever want to describe, usually there's a 3-mark question, right? Agree? So let's see. 1, 2, 3. First one, Yunxun, can you tell me? If you ever want to describe phagocytosis, what's the first one we always need to write for exam? Or formation of your phagocytosis? Huh? Sorry, continue. Again, this is a 3-mark question. So what's the first 3 things that we always have to write in exam? What do you think this protrusion is called? Is it in your exam, in your syllabus to know? So what's that protrusion called? Pseudopodium. Formation of pseudopodium or what? What are you talking about? You don't just say words like here. Doesn't make you so it doesn't make you correct. So first of all, please take note. You are expected to know this is called pseudopodium. Formation of pseudopodium, what's the first key phrase that we always write? Engulf. And place this, what is this thing called? Lysosome. No, this is not a lysosome. You have your pathogen here, and then you engulf during phagocytosis. This is called a phagosome. Okay, so you engulf and then you place your pathogen, okay, in your phagosome. This is so going to be the first one. Whatever I just said, are always key phrase, okay? Because you already seen already. In exams, you always must talk about the word pseudopodium. They always like to bold and underline. They also like to bold and underline your this phagosome here. So the first one is that you engulf and you place your formation of pseudopodium, my bad, okay, which will engulf the pathogens to place it in the form of phagosome. Next one, can you tell me, Chloe? If I were to sketch up, I'll just draw here. Because inside here, what do we also have? What is this thing that we actually have here? What is this thing called? That eventually is supposed to kill this one here. Wait, this is lysosome. So you tell me what's the second thing that will happen during phagocytosis? You actually, side F, you must actually talk about this. Number 2 is what? You will? Okay. What is this called again? And then, what is this called? And then they undergo what? Fusion. Yes, so second one is fusion, okay, of phagosome with lysosome to form what? Can you tell me after we've actually fused, Chloe? The name very generic one. So you fuse, you form what? When you actually fuse, you form what? Together it's called what? Phagolysosome. So please remember, okay, it's in your syllabus to know. So, however, Megan, how do you end off with? Third one is what? Species. Okay, so hydrolytic enzymes and reactive oxygen species will actually and then digest and kill, okay, this pathogen. So I repeat, there are always the 3 things. First one, Yunshin, how do we actually say? Or how do we have phagocytosis? Formation. Don't tell me words.
Pseudopodium to engulf and form a phagosome.
For engulf what?
Engulf the pathogen.
To form?
Phagosome.
Okay, second one. Chloe, how do we say, for exam? So I said phagocytosis is very important. They always like test this. So it's what? With? To form? Next one? Hydro? And reactive? Will digest the pathogen. Understand? So please remember phagocytosis. Next one, I already said the last internal defense. Usually phagocytosis is the most important one because phagocytosis will always help to form what we call your APC. APC stands for what, Myra? Antigen presenting cell. So later we'll talk about that. Okay, so again, essential to form your antigen presenting cell, okay, because without that you cannot bring about your adaptive immune response. Please take note, this is related to this during antigen presentation. Later I'll talk about that. Okay, now again, next one, the last one that brings about this internal defense is your NK cells. Natural killer cells, I repeat, they are the only lymphocytes part of this innate immune response, and they are responsible for undergoing apoptosis of those cells that are already infected or even cancerous cells. They actually secrete 2 types of enzymes, okay, that is almost the same as cytotoxic T cells. Do you know what are they? You know what I'm talking about? NK cells actually secrete 2 things that will help destroy infected cells, okay? Or again, for example, they also help to destroy any cells that, any cancerous cells or any cells that is already infected by pathogens. What are the 2 things just like cytotoxic T cells? First one is called what? Perforins. Please take note, perforins is to increase permeability of these cells that are infected, alright, or cancerous, so that you can allow granzymes, okay, to actually be able to enter your this cells because granzymes are the one that hydrolyze proteins, and that's why that will induce apoptosis. Understand? These two are the same as your cytotoxic T, yeah? Okay, so I already said this one is supposed to bring about antigen presentation, alright? I said already in syllabus, later you can see, right? Once you actually destroy your this pathogens and you form your this antigens, alright? Sometimes all this, let's say macrophages, they can actually present your this viral epitope or any epitopes on my this surface of your this APC cell. I said already, in syllabus, you don't need to know what is the class of MHC, but you still need to write MHC peptide complex. I repeat, this entire thing, because this is actually MHC. So this entire thing is called MHC peptide complex. But if some schools make you memorize what is class 2 and class 1, just tell yourself, for class 2, that is for your this macrophages that presented your this viral epitope on the surface. Alright? Class 1 is infected cells. So it's different, So I repeat, if it's MHC, this one is MHC class 2. Alright? Because this is actually a macrophage. MHC class 1 is for infected cells. They are both capable of antigen presentation. Alright? This entire complex, I repeat, is in your syllabus to know, because we already mentioned, if you never write MHC peptide complex, you will be penalized. Clear? Next one. Again, since we already talked about your innate immune response, we already know how it can bring about your adaptive immune response. Later, I'll talk about the way we typically write. Let's discuss first, because adaptive, if you can see in the summary slide, I gave you again 4 different types of adaptive immunity. Please take note, you are expected in syllabus to know the difference between active and passive, and then your this active and passive, and your this is artificial and natural. Alright, you all should know already. I'm not going to go through in detail, but I'm going to talk about the differences between active and passive first. I'm going to erase. Let me erase first so I can have space. Sorry. Then I'll send you the recording. Okay. For your adaptive, first thing I want to talk about, because we have 2 types. So this is actually to protect and prepare the body for any secondary response, or even to actually eliminate whatever pre-existing pathogen that is in the body. So we have 2 types. First is actually called your active immunity. The contrast to active immunity is passive immunity. This is not found in your notes, so please actually find some space to include in your notes here. So the first difference between active and passive is the involvement of your this T or B cells. Okay, so please take note for active, right? Immune response involves clonal selection and expansion. Okay, so of your this B and T cells. B and T cells. They have both clonal selection and clonal expansion. Please take note, passive immune response does not involve T or B cells. Hopefully you know what is clonal selection. Clonal expansion is your mitosis or proliferation. Clonal selection is whenever you have complementary binding. For example, if I were to have, this is a naive T cell. So if I have a naive T cell from here, you can see we have a receptor that have complementary in shape to your this viral epitope, alright, during antigen presentation. So this is the first difference. Second difference is the source of antibodies. The source of antibodies from your this active immunity, they are synthesized by plasma B cells. Whereas here, the source of antibodies are acquired from either the mother or maybe injection of antibodies through the artificial method. Remember, the acquired from mother, this is like your natural, okay, passive immunity. Natural passive immunity, again, take note, is like, for example, it can be across the placenta or through breastfeeding, so you can actually obtain those antibodies here. We also can obtain antibodies, okay, through injection, okay, from an external source. Another one is difference between active and passive is the durability of this immune response. For your this active immunity, you should know already, since your body is capable of producing all these T and B cells, alright, is definitely long-lived in contrast to this short-lived effect. Because this one comes from an external source. Eventually, this one will undergo degradation. But your immune cells are not equipped, alright, to synthesize those antibodies that can recognize this viral epitope here. So that's why, again, this one is going to be short-lived. So again, durability, we know this is long-lived effect of your active immunity. This is going to be short-lived. Keep in mind, you should know already, I even will be able to synthesize my memory cells. And then later, let's hear later the standard answer for your this memory cells. Another difference we also can talk about is the lag time for the immune response, okay? So please take note, for this one, the immune response for active immunity is never immediate. Sorry. So there will definitely be longer lag time. Okay, whereas here your immune response, okay, for your this passive immunity is almost immediate. So again, shorter lag time. Next one, hopefully you know, memory cells. Memory T, memory B cells are definitely going to be produced. This one, hopefully you know, memory cells are never ever produced. Let's go through as a revision first before I talk about the last one. For your this memory cells, we mentioned before. Memory cells, let's hear. Yunxin, what do we say? And I said, right, sometimes this is a 3-mark question, right? There's a standard answer for whenever we describe for the row. Wait, my eyes here. Every time we describe the row for your this memory cells, what do they provide? Keyword is always what? Immunological
memory.
So they always provide immunological memory. And then, don't stop there. Oi. So it's what? So when you provide immunological memory, how do you end off? For faster? What kind of response? Secondary response, okay, when exposed to the same exact pathogen. Haven't finished yet. And then to form plasma cells in order to produce higher concentration of what? Antibodies. Do you stop that to prevent the person from feeling
Sick.
Yeah, I already said these are the 3 things we always write, and I show you those who are with me before. You already can see I gave you different schools. Answers are always the same. Never ever lose marks if you know what is the role of your this memory cells. First one I want to see is always this keyword: provide immunological memory. Alright, second thing we always need to take note for faster. Okay, secondary response to the same pathogen or antigen. I'm just writing keywords, you all should know by now. So, in order to form higher concentration of antibodies, alright, so in order to prevent the person from feeling ill. So I repeat already. This is always the 4 things, but I said this is usually a 3-mark question. We always write this, okay? Now, so that's why this one, because of this as well, that is why this one is also a long-term or long-lived effect. And Shin, what's the 4 things? Provide
immunological memory.
And then
Secondary
faster.
secondary response.
And then to the same and to another antigen or the same antigen?
The same antigen.
Same antigen. And then by producing
More antibodies.
so that the person
Will not feel ill.
yes. Understand? Last one. The last thing that shows again, okay, the difference between your active and your passive immunity is again the removal of antibodies from circulation. First one, how do we actually remove antibodies from circulation? Antibodies are never removed. But antibodies here eventually will be removed. Okay? So antibodies produced are never removed, are not removed. Because it's produced by their own cells. The own cells will not deem the antibodies as foreign and induce an immune response to destroy it. But here the antibodies come from an external source. So antibodies will definitely be removed. Okay? So remember, you're active, we have different as long as you see, because there are such things that 4 things, right? Whenever you see active, I repeat, active always involves, okay, the body's ability to make this antibodies. Alright? Passive is again, this antibodies come from an external source. This is the main thing that you actually identify, because sometimes they want you to identify what kind. But you cannot just say active and passive. You must further elaborate. Is it an artificial active or natural active? Artificial passive or natural passive? So as long as you see the word artificial, that means it comes from an external source. Understand? Passive with, this natural means that it's naturally, again, inherited. Like for example, natural active is like what? You are infected with the pathogen directly. So you, again, bring about this immune response. Clear? Clear? Now, but just now I talk about is this primary and secondary. What is actually the difference? Because I realize some people do not know what is primary immune response, do not know what is secondary immune response. Can I erase this? Anna? Okay. So I'll just do a table one. You should know by now. I always do a table. There are just 3 main differences to help you recognize, okay? Primary immune response is whenever it's always a first hand, okay? So one thing that we can actually see for primary immune response, sometimes for graphs or tables they actually draw out for you, is that they will always have a longer lag time. So lag time is definitely longer, or you can say delayed, okay? Or you can say slower antibody secretion after antigen exposure. Alright, so we always have longer lag time. Idea is, okay, slower antibody, because this is the first hand, alright, that first time your body is actually exposed to that particular antigen. Okay. So, whereas secondary, things that you look out for graphs, because sometimes they will ask, is that this one will always have faster secretion of antibodies after antigen exposure. Usually it's in a matter of days, can be within days, like 5 days, for example. So it's much faster in response. Okay. somewhat. I purposely put here so you have an idea what it means by faster. Another one is the amount of antibody or the magnitude of the response as compared to a secondary immune response. So hopefully you know the amount is definitely lower. And it's definitely quite obvious if you ever see in graphs, alright? This one definitely you should know, higher amount antibody, stronger immune response. Okay? Last one you can actually look at in your graphs is the effect of your this immune response. You should know already this is definitely short-term effect. Okay, this one is a prolonged effect. So you can see, right, this one is usually in a matter of days, okay? But this one, you can see how your antibodies production, right, and remain in the body for very prolonged periods of time. Okay? So hopefully you know, so usually we'll say this when it's first exposed to the first antigen, alright? But this one is when the same antigen is re-exposed, okay, to this person or individual. Okay, so now we have an idea, alright, what is the difference between your active and your passive. How are you going to describe, okay, our typical innate response is always related to adaptive. And those who did the practice paper that I give out, I said already, one of the answers that I show you, I even star for you for those who submitted, okay, that is always the standard answer we always write. So what is always a key phrase that we always write for exam? So I always want to see is, because you always, when you want to say how the innate will bring about your adaptive, such that you can produce, okay, your these antibodies. So first one, you always must mention about your antigen presenting cells. In exam, you must always spell out, you cannot write APC, you must spell out the whole thing. The antigen presenting cells, okay? Then you write your APC, by the way, alright? Please take note, antigen presenting cell, I already said, can be, for example, in your syllabus, the very common one is like your macrophages. Just now we say macrophages secrete so many things, right? So macrophages also perform as an antigen presenting cell. Dendritic cell are also another type of antigen presenting cells. Okay, so the antigen presenting cells, hopefully you know, we'll always say will undergo phagocytosis. I'm just going to underline the keyword. Megan, what's the next thing we always write? Phagocytosis and present. Keyword is always undergo phagocytosis, then what? Because you want to present this, I want to form this. So it's what?
Present antigen.
Process antigen. So they will process and present the antigen, okay, as what? Keyword you always write in exam is peptide MHC complex. MHC peptide complex, both acceptable. I'm just purposely write like that so you know both are acceptable. I'm trying to say is that in exams, you always need to showcase to me what is the first cell. I don't care about whether you want to have your this phagocytosis of your B cell. Whenever you want to talk about even vaccines, how vaccines can also help you produce high concentrations of your antibodies, always must first talk about your antigen presenting cell. And I always want to see this keyword, phagocytosis and peptide MHC complex. I really say you don't need to know what class of MHC that is, alright? But you still need to write this. I guarantee you it's always a bold and underlined thing. Second thing, we always start off after your this antigen presenting cell. Who is the one that will actually recognize this? You always start off with the word naive. Naive, that means they are not yet matured. Alright? They only get matured after clonal selection here, alright, that will secrete your this cytokines. So after which, I always want to see naive T cell. So the naive T cell, okay, you must tell me, yeah, let me write down first. Keyword is complementary in shape. 2. So the next cell you're always involving is your naive T cell. Naive T cell can either be, usually we will talk about your this CD4, which is going to be your T helper cell. I must see the keyword complementary in shape to what? This is always bold and underlined all the time. And then, okay, this naive T cell, after which, is actually your APC that will actually secrete cytokines. The cytokines hopefully is the one you always write, activate, okay, and undergo clonal expansion and differentiation. I repeat, you always have to write clonal expansion and differentiation side by side, okay, all the time. So, to activate. Naive T cell. And keyword you always say, undergo clonal expansion and differentiation, so you become much more specialized. Alright, to form helper T cell. Keyword is always this, clonal, activate. Clonal expansion and differentiation are always the key phrase to form helper T. Okay? After which, we always say how this helper T is the one that brings about the formation of cytotoxic T, but we always like to elaborate based on your plasma cells. So, okay, whenever we have clonal expansion and differentiation, usually, even though this is the main one, please take note, this one can also form your cytotoxic T as well as your memory T cells. Again, memory cells, let's try. 1, 2, 3. First one, how do you say memory cells? Provide immunological memory. After which, what must we say, Myra? For faster secondary response. In order to faster response, secondary response to the same or different pathogen. Same pathogen by producing what? And antibody, so that to prevent the person. Yes, this is always the way we write. After which, this one, we always need to describe, right? So helper T right now eventually is the one that will secrete cytokines to activate specific naive B cells. Keyword again. To undergo. Keyword must always write clonal what? Yunshin?
Clonal expansion and differentiation.
Yes. Always write them side by side. And differentiation is always what we write. This time is to form what? To form plasma B cells. Keyword, this time helper T. You say secrete cytokines, okay? You always tell me is to activate your this naive B. Keyword is always clonal expansion and differentiation. So this time you are trying to form your plasma B. I'm not going to write because you all should know already, which will secrete your antibodies that will target this antigen for removal. Okay? So for those who do not know, I repeat, these are always the three things we take note. If you want to say how your innate will bring about your adaptive. We always must start off with your antigen presenting cell and how your helper T cell is actually formed. Those that are actually underlined is always bold and underlined because they are key phrases examiners are looking out for. I repeat, this is a marked down process. So if you do not have those key phrases, you're just simply going to be marked down. Understand? So this is actually your adaptive immune response. But I already said in your this summary slide, I gave you your this three main adaptive immune response. We have the cell mediated. Just to let you know, this is again how you have your antigen presenting cells. You can go and look. I already sent to your respective group chat. So your this antigen presenting cells, remember we need to undergo phagocytosis. So phagocytosis is here, right? 1, 2, 3. Describe phagocytosis. Formation of what? What is this thing? Start with P. My eyes here. So continue to place the pathogen in a, or engulf and place the pathogen in a. You engulf, so what's the vesicle cord that contains your pathogen? This is a phagocyte, so it's called a phagosome. Megan, quick. Fusion of phagosome with lysosome to form phagolysosome. Then, and reactive oxygen species will die. Just and destroy pathogen. Okay? Now, do you see eventually we have an MHC because this is an antigen presenting cell. The MHC will actually form this. What is this thing called that has the viral epitope or epitopes on the surface? I'm talking about what is the complex called. Yes, this is what you write for exam. Understand? So, again, this is clonal selection. Clonal selection is as long as there is complementary binding between a receptor and your this epitope. This is called clonal selection. Alright? So that's why do you see secrete cytokines that will cause this? So you always cause clonal expansion as well as differentiation. That's why you can form more T helper cells. Alright, as well as cytotoxic T as well as your B cells. Understand? So this is your cell-mediated response. So what else? We also have your same thing, cytotoxic T. Why do we need to secrete cytotoxic T? Cytotoxic T help to remove any infected cells from the body. Because we have pre-existing viral load, we want to decrease the viral load or pathogen load in your body so you can actually recover from the illness here. Alright, or infection. Cytotoxic T secretes what two enzymes? And what would they induce? Sorry? Perforins and? To induce? Start with A. This is always what we write for exam. Remember, always end off with that. Understand? Next one, we also know your this also have my plasma B cells. So let's go through. Plasma B cells I already mentioned. They will produce antibodies. Alright, later we'll talk about how they actually produce antibodies, but it's actually for humoral immune response. Humoral immune response, hopefully you know, is to block infection because antibodies perform these three things, right? So let's hear. Antibodies perform what three things? 1, 2, 3. First one is what? Oh wait. True. Wait. What the antibodies do? You need to know the three effects of antibodies. First one is for what? Yeah. Like N, like acid and base. Okay. How am I going to talk about neutralization? Neutralization, if you're not sure, you can just write. Neutralization, as you can see here, is when you bind your antibodies, okay, to the toxin. You prevent the toxin or your antigen or your pathogen from binding to host cell receptor. So you can see here, imagine this is actually this virus like influenza. I bind to every single hemagglutinin on the influenza. Isn't that preventing your hemagglutinin from binding to sialic acid? This is an example. So if you do not know what neutralization is, I just repeat that. It's just binding of antibodies to the pathogen or antigen to prevent pathogens from binding to host cell receptors. Okay? Second one is what, Megan? Opsonization 是什么? You mark the Yes. So, but that one is the process, right? So, opsonization, if you want to learn how to describe, especially for your essay, okay, it's like the binding of your this antibodies to mark the antigen or pathogen for phagocytosis by macrophages. So I repeat, if you want to know what opsonization is, it is like purely binding of your antibodies. Once your antibodies bind, it will mark, okay, the pathogen or antigen for phagocytosis by macrophages. Okay, so this opsonization, just now what Mayo was talking about, antibodies, if I were to draw out the structure, we always have heavy chain and light chain. So this is your heavy, you can tell it's a primary, secondary or tertiary protein, Myra? Tertiary, your head, quaternary. So, again, quaternary, again, is because when you are made up of 2 or more polypeptides that are chemically bonded together. Tertiary protein is still within a single polypeptide chain, only one. Now, we have your Fab and your FC region. By the way, I realize some students do not know what Fab and FC actually stands for. Myra, do you know what Fab stands for? No. Fab stands for your this fragment antigen binding region. Okay? Fab stands for fragment antigen binding region. Usually, as you can see, it contains my antigen binding sites. These are the 2 that actually binds to my antigen. Okay. Your FC is actually stands for my fragment crystallizable region. Okay? That's for essay lah, Alright, so hopefully you know this one, FC region, location here, when your this antibody binds to an antigen, you're supposed to bring your this pathogen to the correct phagocytes for phagocytosis. Because you should know I have many phagocytes. These are not the only phagocytes. We have neutrophils, basophils, eosinophils. Okay, different phagocytes is to target different type of your this pathogens. Okay, so that you can actually undergo elimination. Understand? So how is because this FC region, usually let's say if I have a FC receptor that is found on a phagocyte, they can help to activate. So maybe this phagocyte here is maybe your neutrophils, okay? So you can bring and eliminate, all right, for their respective effector function, all right? So opsonization, just to give you a recap again, stands for what? Opsonization is when you have my binding of your antibodies to mark, all right, your this pathogen for phagocytosis. Number 3. The function is what, Kalentia? So we always say is activation of complement proteins. What is activation of complement proteins, Kalentia? So what she mentioned, if you do not know what is this, activation of complement proteins just means trigger the formations of pores, all right, in the cell surface membrane of pathogens, okay, to bring about osmotic lysis. So I repeat, triggers formation of pores in cell surface membrane to bring about osmotic lysis of the target cell. So it can be a bacteria cell. It's called complement activation. Okay, now same thing. You should know already all this. Eventually, the last kind of adaptive is your immunological memory. I know you mentioned already. You all should know what's the standard answer for immunological memory. Okay, so that one is your immunological memory that's generated by your memory T and B cells. Okay, clear? So hopefully you know the purpose is actually to bring about faster secondary response. Faster secondary response has 3 characteristics. What are the 3 characteristics I can see here? 1, 2, 3. First one is what? What did I say? Just now I talked about. This is primary, right? This is secondary, right? So you have?
What does this even mean? What does this graph even mean?
This try to show you primary immune response.
Oh.
This try to show you secondary immune response. So this y-axis your immune response against time.
Okay.
So I already said, because sometimes they will just give you a graph. So what are the things that we can see here?
Higher antibody.
So we have faster antibody secretion. Sorry, higher antibody secretion. Second one is what? Shorter lag time. Agree? Third one is what? Is long-lived or short-lived? Long-lived for secondary. Understand? Clear? Okay, now I want to talk about this antibody. I'm going to erase her. So now you all know how to describe already. How we relate innate and adaptive. You are next expected in syllabus to know this antibodies, structure and their respective function. This will be a recap for some of you which I already went through before. But nonetheless, we still need to know what is the purpose of all this function to help you in your understanding. Let's talk about the first one. These antibodies, alright, they have again a quaternary structure, which is your heavy and your light chains. Now, how do we actually synthesize, all these heavy and light chains? Hopefully you know, we always start off, so heavy is all those long ones, the light is actually this ones. Your heavy and light chains have variable regions as well as your constant regions. They are all encoded by what? This gene locus, So what happens is that we always have a particular gene locus. So, and a particular gene locus to make up one heavy chain. So you can see this is one heavy chain here, the long one. Heavy chain is made up of this red, this green and yellow. Red, green, yellow represents VDJ. So if a gene locus, at a particular gene locus, we actually have many different types of VDJ gene segments. But to make just one variable regions of the heavy chain, you choose usually 1V, 1J, 1D gene segment. So that 1D, 1V, and 1J will actually be rearranged to form a VDJ exon. And that exon will help you synthesize this variable region of your heavy chain. Same thing, the variable region light chain, as you can see, is VJ. It's much smaller, only red and green represents VJ. So at a light gene locus, right, you also have many different types of VJ gene segments. We only need one to make out one VJ, exon to code for the variable region of this light chain. So this is usually how we code for, all right? But let's talk about these antibodies. How do we know the function and what is it related to the structure? So first one, we can see. Some of you, Myra, can you tell me right now? The first thing we can see for these antibodies is we have my FAP region, right? So what is the purpose of the FAP? So it binds so it forms the antigen binding site to bind to what? Antigen. So idea is that will it be complementary in shape? So the structure feature is the FAP region that forms your antigen binding site. It's complementary in shape. Once it's complementary in shape and binds, what is that called? This is actually hopefully you know, it will bring about the process called neutralization. So it's complementary in shape to this viral epitope or epitope of the antigen. To epitope. Please take note, I realize some students do not know what's the difference between epitope and antigen. Megan, what's the difference? This is a subset of these. I can have an antigen But antigen, remember, are like proteins, right? You have different sites of your proteins. Okay. Sometimes any receptor can bind to different epitopes. All this are just different epitopes here. Okay? But I want to repeat your antigen binding site, when they bind, usually we can use, they bind to antigen. But every different antigen binding site recognize a particular epitope. Okay? So we cannot recognize all epitopes. This is a type of epitope. Understand? Okay, so again, it's complementary in shape. So what's the purpose of this? Function of this, you should know already, to bring about neutralization. When you can undergo neutralization, purpose of neutralization is we bind, right? Okay. Purpose of this is to bring about neutralization. Okay. Next one. We have the FC region. So the FC regions of your antibodies or your constant regions of your this heavy chain, they have a conformation. That is complementary in shape. Okay, to the Fc receptors on phagocyte. I already showed you what's the difference between Fc region and Fc receptor. Okay? So they are the ones that bind to Fc receptors that is found on phagocytes. Purpose of this, you should know already, is to bring about opsonization, where you actually target and market, alright, but to promote or trigger phagocytosis by your phagocytes, okay? So it enables opsonization. Next one. Can you tell me, Kalandia, what are the structure features we have for your antibodies? You notice how, again, it's a quaternary structure, you need to have agar interaction. What agar interaction do we have? Disulfate. Is it covalent or is it intermolecular phospholipid attraction? Covalent in nature. Where do you think this is actually synthesized? At which part of the cell? Is it RAR? Golgi. Understand? Golgi apparatus. So we also have what we call disulfide, alright, bridges between heavy and light chains. What's the significance of having this, Valentia? To bring about? Guess what? Because covalent in nature, easily broken. So you guess? Yes. So you give stability, okay, to the quaternary structure. Okay, next one, can you tell me? What are the structure features I have for my antibodies?
The hinge region.
Hinge region is for what?
Is for the binding.
Of how
The
many?
antibody to two antigens.
Okay, so purpose is for what? It gives.
Like flexibility.
Yes. So we have presence of a hinge region. Hinge region actually gives flexibility. Okay? When binding to antigens. Next one, Chloe, what else do we also see here? How many antigen binding sites do we have? 4. 2. We have 2 antigen binding sites. Okay. In my this immunoglobulin, usually for your this class class M, alright? By the way, what's the difference between an immunoglobulin M and immunoglobulin G? Yeah? Because you always use and see the term, right? Immunoglobulin is Ig, yeah? You have immunoglobulin M and immunoglobulin G, right? Agree? These are always the common ones, right? What's the difference? Then M don't have. M also have 2. I tell you, this is always the first class of antibodies, okay? Usually when you are first synthesized, and this is before antigen maturation, okay? So, I repeat, this one, how do we actually become from this to this? Via class switching. Class switching is when you actually, okay, change during your antibody maturation. So you can now have a very specific, okay, function of your this antibody. Alright, so usually you have 2 because it enables simultaneous binding. To 2 antigens or epitopes. Alright, to actually bring about agglutination. Agglutination is clumping, remember? So imagine now I just clump them. This will actually help to increase rate of phagocytosis. Since it allows or enables better clearance by phagocytes, you're better able to recognize a clump of pathogens rather than pathogens that are not clumped together. Okay? Last one you should know already, just now I already mentioned, is your constant regions. Constant regions of your this heavy chains, not light now. Okay? Constant regions and heavy chains are the ones that enables class switching. So it determines the type of class. When I say class, it means the effective function of your antibodies, alright? Because remember, we have different type of pathogens. We have viruses, parasites, fungi, okay? Or even your bacteria, etc. So you definitely need different functions, okay, to target different things. For example, I give you a thing. Doctors, there are different specializations in medical fields. We have gynae, we have for those who are the eye, those are the bone, etc. Right? Pancreas, okay, etc. So that's why we have different specializations. So this is a structure. How is it related to the function? But let's ask. Whenever we have here, notice how one single antibody is capable of recognizing, okay, up to 10 to the power of 12 different type antigens, which is actually a broad array of your this epitopes or broad array of my this antigens, right? So what enables them? Something I need to point out is a lot of students always think, okay, antigen your this variable regions, right? How can I have your this variation in my variable regions? Variable regions again is those in orange on the board, okay? So how will I be able to form different types of specific antibodies that can target your this antigens? Is because of somatic recombination as well as your this somatic hypermutation. But I realize some students do not know what's the difference, alright? Please take note, somatic recombination spans 3 things. I'm going to erase now. Like how do we form like this one? Alright, first one, somatic recombination. Second one is somatic hypermutation. Somatic hypermutation is the one that will bring about what we call antigen maturity. Please know what if that means, okay? So you have to, sorry, affinity maturity, my bad. The idea when I say affinity maturity, let me just write first. This is always going to bring about, okay, this affinity maturity. The idea is that all these are the two main mechanisms that help to increase the variation of antigen binding sites. But let's go through what is somatic recombination as well as your somatic hypermutation. Somatic recombination I mentioned before. I don't know whether you all recall. For some of you, we have again 3 things that will bring about somatic recombination. First thing that will bring about somatic recombination is what we call your germline diversity. This is the least important, but can still contribute. Later I'll talk about that. Second one is what we call combinatorial diversity that comes with 2 things. Third one is called junctional diversity. So what are these 3 that can actually bring about, okay? Now, I already said this is already, take note. Oh, this is actually not, I didn't talk about the germline diversity. Remember I mentioned is that we have many different types of VDJ at the gene locus. So let's say if I want to make a particular antibody, okay? So a particular antibody, let's say your light chain, we have different number of VJ. So light chain I have different number, but I need to mention, okay, V and J are the ones that make up this variable region, but different people may have different numbers of V, different number of J gene segments. Maybe you only have 10, okay, V, but she only has like 6 V. So do you see, right, again, the different number of the multiple copies that you differ from one person to another, that brings about germline diversity already. Agree? Based on your P and C that you learn in your math, okay? Now, again, but the second one is the more important one, your combinatorial diversity. Combinatorial diversity is based on 2 things. Based on number 1, your different combinations of V and J. They are combined together to form a axon. Later I'll explain. Okay. The other one is your different association of light and heavy chains. I'm going to give you an analogy so you can know when I say this 2, then you at least know what you mean. Like for example, you all learn in O-levels your reproduction in humans, right? So reproduction in humans, do you agree? Just a single individual, I can form a variety of different gametes, right? Your gametes that you make are not going to be identical to each other, what? Because you learn meiosis takes place, right? Agree? This is what it's trying to say here. I can form different types of your variable regions of your light chain. I also can form different types of your variable regions of this heavy chain. This is what it means. Analogy for this is like what? You learn in your gametes, male gametes and female gametes will have random fertilization, right? That random fertilization will also increase diversity, right? So this one, your light and heavy chains must associate with each other, right? But there are different combinations of light and heavy chains that associate with each other. So would I have different combinations of variable regions that will be forming this antigen binding site? So this is the analogy for this. But let me go through what I mean. When I talk about here, for those who do not know what it means for your combinatorial diversity, Okay, combinatorial diversity, you can write down is that, first one to explain this, is when you form different combinations of V bracket DJ gene segments, okay, to form variable regions. Please don't write different combinations of genes, it's different combinations of gene segments, okay? You don't also write different combinations of regions, you will be penalized. So the idea is that they are going to be rearranged and then eventually they will form an exon. So I repeat, different combinations of V, D, J are rearranged to form VDJ exon. I repeat, rearrange together to form VDJ exon to code for variable domain of heavy chain, to code for variable domain of the heavy chain. Same thing for the light chain. You also say the next bullet point, you can also write VJ gene segments rearrange to form VJ exon, to form VJ exon for variable domain of light chain. So let me give you an example. Let's say this is my heavy chain. Within each one, there are multiple. So usually, there will always be recombinase enzyme that will remove, and then it causes the rearrangement here. So this one here will now form the VDJ exon. Eventually, you can undergo translation, and you can code for, again, the variable regions of my heavy chain. Okay? The same goes with your light chain. So this is your first one, right? This is the one that you can form the exon. Okay, second one is, what is this? So this one, again, please take note, is that we have further combination diversity. So this is when you have the binding sites, because you have different light and heavy chains, okay, domain that combine to form different antigen binding sites, all right, which can bind to different antigens. So I repeat, if you do not know what it means. So different association of light and heavy chain variable domains, okay, help form different antigen binding sites to bind to different antigens, generating different antigen specificity. So I repeat, association of different light and heavy chains, variable domains, form different antigen binding sites to bind to different antigens, generating different antigen specificities. Okay? So this is what we have. The third one I say is junctional diversity. Remember, we need to choose 1V, 1D, 1J gene segment, right? So eventually I can form an exon. So to choose one is that you need an enzyme called recombinase. Recombinase is an enzyme that will cut and ligate. So what happens is that sometimes when you actually cut and ligate, this has non-specificity. That means the cutting and ligate, sometimes you can actually cut within some region, some nucleotide sequence of a V gene segment, for example, and then you ligate. Or maybe you have not cut properly, you add other gene segments that is just outside of your V gene segment. So isn't that can result in loss or, again, addition of nucleotides? The entire nucleotide sequence will be differing already, right? If your entire nucleotide sequence differs, wouldn't that also code for different amino acid sequence and therefore different polypeptide chain that will make up the variable region? So junctional diversity, for those who do not know, it means that, again, nucleotides that are added or lost during recombination to form different antigen binding sites. Nucleotides are added or lost at adjoining sites during recombination to form different antigen binding sites. Okay, hypermutation. So what is hypermutation? Hypermutation, right, usually will bring about what we call affinity maturation. Okay, so this one usually occurs after many rounds of your what do you call that? Antibody that is exposed to the antigens already. So you want to undergo mutation that will eventually increase the affinity of the antibodies to the antigens. So the first one you can write down if you do not know what somatic hypermutation is. But I need to point out, somatic hypermutation doesn't occur here. It occurs once all the VDJ units are all rearranged already. That means it's already at this level, the last stage just before translation. So for hypermutation, you can write is occur at rearranged VJ and VDJ exons, okay, of light and heavy chain gene loci. So occur in rearranged VJ and VDJ exons of light and heavy chain gene loci during clonal expansion of B cells. During clonal expansion of B cells to change conformation of antigen binding sites, to change conformation of antigen binding sites, to increase affinity to antigens, and that is affinity maturation. Okay? Because usually when you say complementary, you still may have low affinity, but what we're trying to do during affinity maturation is to change it slightly in conformation so you can bind more snugly towards an epitope. Okay? So this is what an affinity maturation means. So these are only the two that will bring about variation in these variable regions. But besides that, we also have what we call class switching recombination. Class switching recombination is what form your this heavy chain, alright, in your this FC. That's why we can have a variety of my FC regions, or your or your constant regions of your heavy chain. So what is class switching recombination? Please take note, class switching recombination occur in activated B cells. That means it's not naive B cells. That's why, right, your first antibody you form is usually your immunoglobulin M. Class switching occurs in activated B cells. Idea is that you want to form different, classes of antibodies. But what is class switching? If you want to know what's the definition of class switching occurs where DNA rearrangement occurs at the constant region of the heavy chain gene locus. So I repeat, where DNA rearrangement occurs at the constant region of heavy chain gene locus. Resulting in different class of antibodies with different effector functions for the same antigen specificity. So I repeat, resulting in different classes of antibodies with different effector functions. Some can be for purpose of neutralization, some can be for opsonization, some can be for complement activation. That's what I mean by different effector functions for the same antigen specificity, because they all still have the same variable regions or antigen binding sites. So this is what class switching is about. Okay? I repeat, this only can occur during activation of your B cells, by your this helper T cells. Alright? Any questions? No. Okay, I know I talk a lot, but let's try. Notice how I gave you two worksheets here. First worksheet I gave, again, is your this MCQ. That one is for your own practice. If you want to revise for the chapter already, the MCQ is going to be the fastest way. Answers is already attached. But I want to look at your structured question so we can actually practice what we just learned, okay, and emphasize. Now, same thing, each one of you, I want you to do your respective questions for me to mark. Shin, Megan, and Palantia, do you see question 1B and 1C? I want you to do 1B and 1C. The rest of you, do you see we have 2B and 2D? Can you help me do question 2B and 2D first? So I'm emphasizing the main things so you can understand this chapter better. Once you're done, let me mark. By the way, for those who want to know what this one in detail, I'll just elaborate. We always start off with this. We always then have this. And then we always have this. From here to here, how do I make from this? Immunoglobulin M, I already mentioned, is always going to be the first class of antibodies that is secreted by your B cells. So after one round of what we call class switching, we will form here this immunoglobulin G. Immunoglobulin G, okay, is when we first form, but usually this one, we have lower affinity. Okay, we already have the respective effector function of your antibody, but after rounds of somatic hypermutation, this will increase affinity maturation or affinity, okay? Especially during the primary immune response. So what I'm talking about is affinity maturation, right? At first is lower, okay? This is usually during a primary immune response. After secondary immune response, okay, binding affinity to antigen will definitely increase, all right? Because during a secondary immune response, you know that, again, you're already re-exposed to the same exact antigen, okay? So in case that you need to understand what is your immunoglobulin M, the G, what's the difference between them? Alright? So please know, because this is always the first class. It's not G become M. Usually concentration of this one is also lower, since this is usually part of your primary immune response. Okay? Usually by the time it reaches the immunoglobulin G of higher affinity or during affinity maturation, that is usually during your secondary immune response. Okay? Again, just try these 2 questions so that I can go through. Remember, for suggest questions, sometimes suggest is with an explanation, so 1 mark for, again, take note, ask yourself. Sometimes the suggest, they also have an explanation. It's still an explain question. So ask yourself, are you using the keyword technique or the overview point technique? There's always these 2. When I say keyword, it also can comprise of like how, okay, the marks can be delegated based on the graphs itself. That means the trends of the graphs. Okay? Once you're done, let me mark. Okay, you always must specify for me. Either because, as you can see, this is an explain question. They are saying product of more than one gene. Okay, we already learned that this more than one gene is, there is no keyword. We have to use the overview point technique. But we know that if more than one gene, you need to specify why is it more than one gene. I cannot accept if you just say one antibody is made of heavy and light chains. It's made of 2 heavy and 2 light. Without the specific number of heavy and light, usually for me as a marker, I will not give you the credit because you should know how antibodies is made of. Second one, you already mentioned, I don't care about the VGJ gene segments. They are saying how come it's more than one gene. So each of your this heavy and light, they are encoded by different or 2 genes. So how many genes are involved? 2, even though there's 4 polypeptide. Do you know why 2? One light can be coding for 2 light chains. One light gene code for 2 light chains. One heavy gene code for 2 heavy chains. Understand? So because you never mentioned about that, I cannot give you the credit here. Clear? Next one, keyword. They say different specific antibodies generated. Oh, okay. So we already learned because of different specific antibodies generated. The word is called specific. Do you need to talk about class switching? So you have to be very careful. No. Class switching merely talks about and determines the effector function of antibodies. Okay? But the keyword, right, they didn't just use different antibodies. They use different specific antibodies. So specificity is always based on what? Binding to the antigens, binding to the epitopes. That's why we talk about somatic recombination and somatic hypermutation. You understand? Notice how the number of marks here is 4 marks. 4 marks, right? How am I going to delegate the 4 marks accordingly? Because now you know, got 2 mark, 2 mark. No. You know why? Because we know if it's only based on somatic recombination and somatic hypermutation. Somatic hypermutation, how many points? 1. Yellow. And then you should know already 3 marks must be somatic recombination. So you can talk about what? Germline diversity, combinatorial diversity. In fact, combinatorial diversity, you can actually extend it to 2. Third one is actually junctional diversity. You understand? So at least maximum 3 marks for that. I'll give you the credit. Please do the other question while waiting. Now, again, that's why you mentioned how somatic recombination results in different VDJ combination of gene segments. Fine, I will accept for this. You code for the light and heavy chains. Idea, this one will lead to what kind of diversity? Keyword is called what? Combinatorial diversity. Elaborate. That's why I wrote there as 1, 2, 3. Please specify the type of diversity will lead to. Also have different, okay, fine. I will accept this one actually because you already mentioned generating different antigen specificities, even though you don't have the word combinatorial diversity. Hypermutation, because rearrange heavy gene segments. Change conformation, fine. You mentioned how conformation of this to increase antigen affinity. I'll give you the maximum 2 marks because you only talk about one type of somatic recombination. Okay, but I only give you 1 mark as well for your this somatic hypermutation. Okay. Who else? Somebody? Let's see. So just explanation for the first 12 weeks.
I don't know why it went down.
Went down because this is your primary infection. HIV has latency stage. That's why it rises again. That's why it can still rise, ma. So the initial decrease is because of activation of your primary immune response. It rises is because of what? There's decreased viral load first, alright? Agree. Or you can say how you have or you can say how, let's say, right, the initial decrease is because the HIV virus is actually killing your this host cells, right? That's why there's an initial decrease. And then there is a rise is because of recovery, right? Because again, how come I may have actually immune response. This is only within the first 12 weeks. So they say primary infection with HIV some more. Okay, So again, there might be actually subsequent recovery. What?
Also, it's not like the clonal expansion thing that's why increased T cell.
No. You already say it's from a primary infection here.
So it's the phagocytosis.
No, you don't talk about. They are not asking you to describe. You always have a feeling that you always need to describe what is the process, but they are not asking you to describe. If they ask you to describe, they will just say what are the process or how will it lead to the increase in antibody production.
So this is
They are
just about
asking you for an
HIV.
explanation what could account for an initial decrease and what could account for the rise. As long as it sounds logical, I just accept. Actually, you need to learn how to see what the question is really asking for you. You have issues about seeing this. They are already saying this is based on the graph. You already know based on a graph, why 2 marks? 1 mark for the drop, right? 1 mark for the increase, right? Do you think it's about description of a whole thing, about secretion of cytokines and stuff like that? Do you think they want the whole description of how you have phagocytosis either? No. But you are writing that.
Wait, then the secretion part is at the back, or don't even have?
You don't even need to have that.
No, like, is it at the back of the graph, or it's also not counted?
Not counted for that. So why it drops is most likely is because maybe the immune response is able to generate. Another one, increase because CD4. What? I also accept if you say that the bone marrow has differentiation of your hematopoietic stem cells to actually form more CD4 cells. I also accept because it's a suggest question. You understand? Suggest means what? Is it based on background knowledge?
No.
It's based on what? Anything that sounds logical, plausible, I will accept as your answer. What? Think about the techniques I mentioned in class. So no. You understand? It's not about the description. Please learn what they are asking you. Begins to rise when CD4 T cell fall below 350 per microliter. So the idea is, because they say several complications. Several complications means that how come your CD4 eventually drops. So the reason why CD4 cells eventually drops is got to do with your they say begins to rise. So first keyword is rise first. Second key phrase is fall below. That's why it's 2 marks. Because this is an explain. Right? Right? Where is your rise?
I didn't see the rise.
So that's why always look at the number of marks. And then you know it's explained already. Always ask yourself, class, I need to repeat again. Whenever you see the command word is called explain, I guarantee you, despite what topic they give you, explain question, there's always 2 techniques. I want you to imbibe this in your head. Are you going to use the keyword technique or the overview point technique? A tip to use your keyword technique comes in 2 ways. Keyword technique, either you look at 2 key phrases in the question that examiners deliberately use, okay? So you know how each mark are delegated to that specific keyword. Second one is, for your keyword technique, you look at your graphs. Sometimes your graphs, they ask you to explain the graph, you look at the number of trends. If it's 3 marks, usually there's 3 trends. You understand? And then last one is the overview point. When do you consider overview point? When it is not the keyword technique lah. You understand? So you mentioned here is the drop. You say the drop is that there is less T helper cells. Less T helper cells, why? Why is there less T helper cells? You never mentioned due to a compromised immune response. You need to mention how come it's lesser T helper cells, because you need to tell me how come can lead to tuberculosis and stuff like that. And then idea is how come you can lead to tuberculosis. You know tuberculosis is the formation of, we have latent tuberculosis and active tuberculosis. Latent tuberculosis is when you form a granuloma. So you're trapping all the mycobacterium tuberculosis bacterium within that, so it can slowly be eliminated by your T cells or your B cells. Then active tuberculosis is when the granuloma undergoes necrosis, that means it bursts. When it bursts, you are releasing the Mycobacterium tuberculosis. That's why you are now infectious. That's why it's called active TB. Latent TB is not infectious yet. But they already say that they can lead to several complications like tuberculosis. They are referring to active TB. You also can talk about how come you can have active TB here. Okay? Again, they are not asking for the process. You are very excited to write the process all the time. But this one is not asking you how come you will have falling below, how come there's lesser secretion of cytokines? I don't care about that because you already know 1 mark is got to do with why there's lesser or fewer CD4 cells. So the idea is that you can tell me how there is loss of T helper cells because of a compromised immunity since HIV target T helper cells. Another one is that, so how come it can lead to tuberculosis, the lack of CD4, prevent the formation of granuloma to prevent the spread? Because this is tuberculosis. I cannot give you a single credit. Who else? Okay, let's see. I will not accept if you just made up of more than one. Okay, reason being is that you are expected in syllabus to know how many polypeptide chain the antibody is made up of. So how many specifically? Then specifically, do you know how many heavy and how many light? So as a marker, I'm very strict because once you know, you never write that, I cannot give you a credit even though you say more than one. To be honest, right, do you know there's only 2 genes that code that 2 heavy and 2 light? The 2 light are identical, the 2 heavy are identical. You just say that it's coded by more than one, so more than one gene is required. I cannot give you the credit because you could have actually explained to me. First of all, this is overview point technique. The antibody is made up of 2 heavy, 2 light. So it's different polypeptide chains, right? In fact, if you just write more than one, I cannot give you a credit. If you write 2 different types of polypeptide, maybe I can still give you the credit. Then each type of polypeptide is encoded by different genes, or you must tell me there are 2 genes that will code for the this protein structure of your antibody. You understand? So I will not give you credit. It's not because your understanding is wrong. You didn't specify. Always give me sharp answers with 0 ambiguity, especially for questions that's 2 marks or less. Understand? Somatic recombination, different combinations. Please always use the term combinatorial diversity. You cannot just say this VD, why you bracket the D? Just now I bracket the D is because I'm considering both heavy and light. If you want to specify in exam, always specify what is the gene segments that code for your heavy, what is the gene segments that code for your light. Rearrange to form this, rearrange to form this. You never elaborate what kind of you never use the keyword combinatorial diversity. That is why I specified that. If you never use that one, I cannot give you the credit either, even though you write somatic recombination. Next one, you say different association of your light and heavy chains to form different antigen binding sites. Because you write different antigen specificity, okay, fine, I can give you a credit, but hopefully you know it's under the umbrella of this combinatorial diversity. You still need to write this one here. So in fact, you should have written further combinatorial diversity has blah blah blah. Nucleotide added coded. What do you mean? What kind of diversity is this? You never mentioned as well, so I only can give you maximum 1 mark. And then same thing, right? Notice how you never talk about this somatic hypermutation. So, Cheryl, one thing I want to ask. Whenever you want to do any question, don't be so excited in just writing out answers, because I already said, always see how the marks are delegated. We already learned if you want to make different specific antibodies, I no need to explain why class switching is not accepted, right? Do I need to? So, idea is, you know already, it's based on the 2. Somatic recombination and somatic hypermutation. So how many marks of somatic hypermutation? 1. The other 3, maximum 3 marks for your somatic recombination. You understand? Okay? Who else? Quick. I still need to do infectious disease. For 6 weeks, decrease from this. Megan, you don't need to quote data. Do you know why? You just need to say decrease. Okay? So, okay, quote data lah, yes, but you don't need to quote specific data for explain. So we say decrease. This is following the HIV infection net. CD4 cells do not activate naive B cells. CD4 cells are actually being killed by two things, either by the virus or by your innate immune response because they are infected cells. That's why it led to apoptosis. That's why you have that drop. You understand? Decrease ma, how come there's fewer CD4? It's not because they are activated. You always must have a pool ma. Clonal expansion will occur. If you activate to form more, right? Agree. So how come it decreased? That one is wrong. Wrong understanding here. You don't want to increase more. Oh my god. Okay. Now you understand. So how come it slightly increased again? Because that subsequent recovery, maybe I can accept a few things. Maybe you have like decreased viral load, okay, because of the immune response. So you decrease destruction of CD4. Or you say that you generate again more CD4 because of differentiation of hematopoietic stem cells. I accept what? That one makes sense because it's a suggest question. You understand? So it begins to rise, secrete cytokines. Why you talk about B cell? Oi, they are asking and focus on rise for your CD4 cell. You don't need to focus and tell me about your B cell. So the idea is that when you rise, if you phrase it in a sentence idea is to activate and have more T helper cell so that you can again activate, okay, and cause clonal expansion and differentiation of B cells. I can at least accept that, which could account for why there was a rise. You understand? But you phrase it in a sense that you secrete cytokines to activate. Okay, fine. I can give you a credit. Fine for this, but you should mention more is to create more. Okay, low this. No, the idea is that how come? Okay, fine. Granuloma cannot form. The idea is you don't even form cannot even form granuloma because of lack of CD4. Granuloma is when you have all the phagocytosized mycobacterium tuberculosis together with T cells and B cells. But lack of this, you cannot even form granuloma. I just need to point out this. It's not they undergo necrosis. Necrosis can only occur when the granuloma enlarge. But you couldn't form granuloma in the first place. So concept is wonky here. Okay? You don't need to quote data because this is an explanation question. You just need to tell me a decrease. You cannot tell me it's a wrong phrasing. It's called infected. Okay, fine. So I accept if you say how this, but you use the wrong terminology. Don't write attack. Wrong terminology. I cannot give you a credit. Okay? I don't care about whether the remaining T cells can undergo clonal expansion or whatever. Idea is that how come there was a rise? It's not because they undergo clonal expansion net. It's because of this recovery. I can accept if you can say how maybe the innate immune response decreased the viral load. That's why, again, lesser destruction of CD4 cells. Or you can say how you have differentiation. So I cannot accept for this one. If you just, instead of writing attack, you write infected, which is the correct terminology, I can at least give you 1 mark. Okay? The other one, 2 key phrases. One is rise, one is fall. So that's why 2 marks, 1 mark for each. You never talk about why is there a rise. So take note about this. Whenever you see explain, I already say, ask yourself, is it keyword technique or overview point? This is a keyword technique. 1 mark for the rise, 1 mark for the fall. You didn't talk about the rise. The fall, you say that causing apoptosis. So it's lesser than lesser this. Okay, so I see lesser to trigger primary immune response. Why is it primary infection? Oh, okay, primary immune response. I can give you 1 mark for this because again falls below. There's lesser this due to apoptosis from the primary immune response, but you can further elaborate. Okay, so I only can give you maximum 1 mark. Okay, come, let's go through. Answer sheet is behind now. But anyway, I want to purposely talk about this because it's data analysis. Alright, for part B, I need to go through the first one. Notice how, right, this one is a suggest question, but they use the word explanation. So even if you have the word suggest, but if you know that this is still an explanation question, ask yourself, keyword technique or the overview point technique? Keyword technique, because there's only 2 trends that you see from the graph. So 1 mark for each. Consciously think how you want to delegate the marks. Okay, don't just vomit out answers because you'll waste your time writing. I already mentioned to your girls is that bio, especially for paper 2 and paper 3, why is it so difficult to score for bio? Because of the limited time. Your brain cells must work like that. So you don't waste your time writing chunks and chunks of answers that is not going to give you credit. I'd rather you take your time and be smart about it. Spend the time wisely in writing answers that you'll be credited for. Okay? So the first initial decrease, answer sheet is behind. But because this is a suggest question, I will accept any logical explanation that will cause the decrease in your this CD4 cells. So if you tell me how, because HIV targets CD4, they will infect CD4 cell. So your primary infection, keyword is a primary infection, your primary immune response or your innate immune response will actually target your disinfected CD4 cells and will bring about apoptosis, causing the decrease. I will accept for that. Or you can say the virus, right, can also kill your this CD4 cells. What? If you say the virus also kills CD4 cells, I also will give you a credit, maximum 1 mark. The other one is how come there was an initial rise. So the initial rise, like I said, I will accept a multiple ones. If you tell me that, okay, maybe the innate immune response because you decrease and kill all the infected cells, right? Isn't that decreasing your viral load? So in other words, would I actually allow for, again, more CD4 cells to actually be formed? Or you can say there's differentiation of more hematopoietic stem cell. You cannot write HSC. In exam, you must spell it out. Okay? So HSC is your hematopoietic stem cell to actually form more naive T cells or CD4 cells. I also will accept. You understand? So I repeat, please always ask yourself, keyword technique or overview point technique? Okay, bio, there is always a technique to score. So you must make it as a habit. Understand? Next one. Notice how it's 2 marks. Again, this is explained. You should know already. This is already keyword technique. You need to tell me why is there rise and why is there fall. So rise, you can tell me, for example, is like what? You have, again, more antigen presenting cells, okay, to actually process, alright, and then present your this viral antigen on your this, bacterial antigen on your this cell surface membrane to actually cause the activation and clonal expansion and differentiation to form more T helper cells. I will accept that. That's why it causes the initial rise. We are not talking about HIV this time because they already say tuberculosis. Second one, how come is there a fall? So for example is, imagine if I have HIV or if I have HIV positive or AIDS, wouldn't that already cause the loss of T helper cell? So you cause the loss of T helper cell is because, again, with lack of T helper cell, hopefully you know tuberculosis is when either you cannot, it's active TB, yeah? That means if they specify tuberculosis here, that means this person is infectious. There are either two things that happens for active TB. Either you cannot form a granuloma. By the way, granuloma is when all your phagocytosized mycobacterium tuberculosis is trapped and contained within this kind of vesicle-like substance with a lipid coat, alright, that's surrounded by all the T cells and B cells inside. So you are containing the spread of mycobacterium tuberculosis. At this stage, you form a granuloma and usually you're called latent TB. So, however, sometimes for active TB, this granuloma can enlarge and burst to release and spread the mycobacterium tuberculosis, or you cannot even form the granuloma in this case because there is no T cells. Understand? That's why it can cause complication. Just tell yourself 1 mark for rice, 1 mark for 4. Alright? Next one, for your B. I'm very particular, even though it seems like a very simple question, I just want to mention, this is using what? Overview point technique. But I'm very strict because you should know there are some protein structures you are expected in syllabus to know the exact number of polypeptide chains. So I cannot accept if you just tell me made up of 4 polypeptide chains or you write more than 1 polypeptide. Because antibody is one thing, you see more than 1 gene, you need to specify. So it's made up of 2 heavy and 2 light, you must specify the number, then I give you the credit here. However, hopefully you know, it's not encoded by 4 genes. Okay, it's encoded by 2 because the 2 light chains of your antibody are the same. The 2 heavy chains of your antibody are also the same. You all understand? So I need to see 2 genes, or you can say each different type of polypeptide is coded by different genes. Okay? Next one, part D. Again, notice how I'm particularly choosing explain questions. So for this one, you ask yourself, keyword technique or overview point technique? Sometimes, again, 4 marks, they already use the term different specific antibodies. I have seen some students talk about class switching as well. Class switching is not relevant here. Why? Class switching does not actually bind or it shows specificity. What? Specificity is always based on your antigen specificity. So that is your variable regions. So how can I form variation or variable domains? Is by somatic recombination, somatic hypermutation. Understand? So take note, class switching going to be rejected here because they never write different antibodies. If they write different antibodies, fine, I can accept class switching. But they write the word different specific antibodies. So very crucial. I already say the way examiners choose to use phrases or words to structure their questions is all intentional. You need to get behind the minds of this examiner. So I need you to take note. You cannot just write somatic recombination. I need you to specify what kind of diversity we have. I need to take note now. Whenever you want to describe your somatic recombination, we usually will use this 2. It's not that I will not accept germline. I also can, but you need to specify. Okay, start this question please. Answer sheet behind. Can you help me highlight the word? It's different combinations, they by the way need to specify. VDJ gene segments of heavy gene locus, okay, and VJ gene segments of light gene locus, leading to combinatorial diversity. I don't want to see just these gene segments. I don't want. I want you to specify both the heavy gene locus and the light gene locus. So again, enables different combination of VDJ gene segment of the heavy chain gene locus and VJ gene segment of the light chain gene locus. Okay, which is missing from the answer sheet. Next one, you also can tell me, do you see they must write the word combinatorial? So highlight combinatorial diversity again. Combinatorial diversity, please always specify. And then they have association of different light and heavy chains. Okay, which will further increase combinatorial diversity. I know there's something missing from the answer sheet. Can you also help me write? Is that junctional diversity? is also established. They don't have that in the answer sheet. So junctional diversity also occurs where nucleotides are added or inserted. When nucleotides are added or inserted at joining sites during recombination to form different antigen binding sites. To form different antigen binding sites of the variable domain. Somatic hypermutation, highlight the word rearrange VDJ gene segments, rearrange VJ gene segments. Without the word rearrange, you will have no credit. Because I already say hypermutation happens after recombination. Okay? Alright, anyway, can I continue? Go back. I need to talk about infectious disease, antibiotics, and vaccination. First one, if you were to flip on your notes, you actually can take note of this viral and bacterial infections. First of all, we need to know what is the definition of infectious disease. If I ask you to define infectious disease, always need to define the first one and the second one, right? So, what are disease and what is infectious? If you ever want to define what infectious means, and what disease means you always need to tell me two things. Caused by a pathogen. Second thing you always need to tell me is that, because this is disease, right? Okay, so you affect or cause ill health or affect the normal functioning of the body. Infectious, you must always tell me how this can be transmissible from one person to another. Okay? In syllabus, we are expected to know 3 types of infectious disease. We'll be going through tuberculosis, influenza, okay? So, first one, I'm going to summarize this. So, we have this tuberculosis. We need to learn what is actually the cause or the transmission of this disease. Okay, so what is the the pathogen involved? Hopefully you know the pathogen involved. Please learn enough. You are expected in syllabus to know how to spell out the whole thing. Mycobacterium, tuberculosis. Alright, first of all, mention for this Mycobacterium tuberculosis. What am I missing there, Megan? Sorry? Yeah, you must underline. Give me a moment now. course, this is a scientific name. Influenza, hopefully you know, we have influenza virus, okay? And this is actually your human immunodeficiency virus. Next one, do take note, how do we actually transmit? So the key thing that we always write is that this is always transmissible, okay? By what? Aerosol droplets. Do not stop here. We always need to elaborate. When and this is always the thing that we write for exam. Infected person coughs, sneezes, or speaks, and is inhaled by an uninfected person. Okay? Influenza virus, we should know, is that influenza can cause flu illnesses. So same thing. Transmissible by droplets of moisture. Okay. From lungs of an infected person. And an uninfected person inhales the droplets. Okay, you're quiet. Hopefully you know this one. It causes AIDS by your human immunodeficiency virus. So it's transmissible by sexual intercourse. Or sexual contact with an infected person. But not necessarily must be this. It can also be sharing of contaminated needles. Okay, or you can say that an infected mother to the fetus. Okay, so now we need to take note how come, and then later what are the effects for this. So this is actually going to be the step-by-step how tuberculosis actually occurs. You need reference. This is actually a reference. So I'll just use this as a guideline first, okay, before I write on the board, so you can at least explain. So usually, also in your summary slide, when I give out, so when you have tuberculosis bacteria is very unique because they are the ones that have this thing called mycolic acids found on the cell wall, on the peptidoglycan cell wall of this bacteria. Okay, mycolic acids are the ones responsible to prevent the formation of phagolysosome during this phagocytosis. Okay, so thereby your bacteria, this one, is not easily killed by mere phagocytosis alone. So what actually happens when you inhale droplets of your Mycobacterium tuberculosis? I'll just use diagram to explain first. You all have this in your summary slide. So idea is that when you inhale, okay, we have alveolar macrophages that will first want to engulf, right? But in the phagosome, remember, bacteria has presence of mycolic acids. This mycolic acid inhibit the fusion of phagosome with lysosome, and you cannot form phagolysosome. So what happens is that bacteria can survive, right? So, however, idea is your bacteria survive and it will continue to multiply within your macrophages. So do you see how we have all this look like the purple and your green ones, and your dendritic cells, the one in orange, all right? They will have all this bacteria, right, that is in the phagosome, right? Do you see the bacteria in the rectangular red symbol? So what happens is that these macrophages, however, will, because the bacteria did not die, right? They know the bacteria was not destroyed. The macrophages will start to secrete cytokines, and then they will try to recruit more macrophages, as you can see here, okay? So they will recruit more macrophages to the infected alveolus, eventually forming this ball-like clustering of infected cells surrounded by these other immune cells. We call this, again, a granuloma. So do you see we have a granuloma? So the granuloma here, okay, at the center, right, what happens is that because you will have this T cell and B cell that also surrounds or is found within the granuloma, so you are still containing and preventing the spread of your Mycobacterium tuberculosis, okay? So you are still not infectious at this stage, okay? We are said to have latent TB. Latent TB, that means you still can recover potentially from latent TB if you are not immunocompromised, okay? So, but however, let's say you're immunocompromised. What happens is that immunocompromised, for example, you have AIDS, right? You have very little, okay, T cell, B cell. By the way, right, bacteria within here, they can undergo binary fission. If the rate of binary fission is equal or more than, less than the rate of killing by your T cells, B cells, you will still have latent TB. That means you still potentially can recover from TB. But let's say you are immunocompromised. You have lesser T cell, B cell, right? Then it means that the rate of killing is now lesser than the rate of binary fission, right? That means your Mycobacterium tuberculosis can divide, and then divide at a rate so high, it will cause your granuloma to enlarge. Your granuloma at the center of this granuloma, eventually when it enlarge, you will eventually burst. This burst is called necrosis that you also see in your notes, okay? On this page, last page of your notes, you have this first and second column in the diagram. Necrosis occurs, is called cell death by necrosis. It involves the rupturing of not only the granuloma, but your macrophage, okay, cell membrane. So you release cell contents inclusive of this Mycobacterium tuberculosis, okay? So usually, take note, TB disease is said to occur the moment the tubercle or this granuloma ruptures. So the bacteria will spill into your bronchioles and spread throughout the lungs. So you are actually leading to what we call productive cough, and then that will further spread to another uninfected person. You understand? So this is what happens and how come we can die from it. Okay, so I know I described from here. I'm going to write down, so at least when you write down, you know what is the step-by-step process. First one, if you want to have tuberculosis, what are the steps that occur once you have this tuberculosis here? Is that alveolar macrophages are the one just now I mentioned already for phagocytocytes. Okay, the bacteria. Okay, however, presence of glycolic acids prevent breaking down. Number 3, again, you need to talk about how the bacteria will therefore not be not dead, right? Okay, idea, let me just further elaborate. Number 3, Number 3, the bacteria can survive and multiply. So when they survive, your macrophages will try to recruit more immune cells to eventually form a tubercle. My space are right at the top. So eventually, you can see how we have cell death, alright, if the person is immunocompromised. And this cell death by necrosis involves your rupturing of this macrophage cell membrane as well. Okay? Number 6 is that eventually your tubercles can rupture. Alright, usually disease may be arrested, we can remain latent for years, alright? But let's say he's immunocompromised, you have TB disease. So when TB disease actually occurs, okay, the tubercles can rupture. Okay, because they enlarge, right? I already told you how we can enlarge, alright? Okay, so the bacteria can actually spill into your bronchioles and spread throughout your lungs. So idea is you're going to lead to what we call the productive cough that can produce your aerosol droplets in order to spread the disease as well. Okay? how it actually affects her. Let's talk about this flu or influenza. So you should know already this one causes tuberculosis. This causes what we call the flu. So what brings about the formation of flu? Again, you should know is by replication of this. But how come this is actually bad? So we already know how TB actually or disease is brought about. But again, we need to take note, how does this actually cause illness in the person? It's because you form new influenza virus, right? New influenza virus undergoes the exit via what process, Yunxun? How does new influenza virus exit the host cell? Huh? What's the process of exit? Come on. By? How new influenza virus exit host cells after the replication stage? Megan? By? Not exo. Yeah, but they cause loss or gain of cell membrane. Loss of cell membrane. Wouldn't that actually, okay, cause your this host cells to eventually die? Okay, so new influenza viruses, okay, but off. Alright, from infected host cell. So when they bud off, you should know how the host epithelial cells will eventually die. So because of the budding off. When they bud off, the epithelial cell lines. The buildup of dead epithelial cells will bring about inflammation. Okay, so I haven't finished yet. Idea is that once you build up, okay, the epithelial layer weakens, right? So that's why any individual is going to be more susceptible to bacterial infections. Okay, AIDS. So what's the effect for this? So for AIDS, right, hopefully you know, same thing, it binds and target your this T helper cells directly, right? Agree? So how come you actually have this? Same thing. HIV viruses, but off. when they bud off, this time the infected T helper cell will eventually lyse. Alright? Because you should know already it causes a loss of cell membrane. Okay? So when it lyse, it means that you have fewer T helper cell. And you should know it's already part of your immune response. Alright? Fewer T helper cell, so immune system is actually compromised. So when it becomes compromised, you should know already more susceptible to opportunistic infections, alright, that can eventually lead to death if your infections become unmanageable. Okay. Oops, sorry about one. Alright, so all this is the infectious disease that's in your syllabus 2 note. Oh, I forgot to mention. Crap. Now let's see how you can actually break down. My bad. 7, I can even further elaborate. My bad. We need to take note when you actually keep on rupturing, right, all your tubercles. When you rupture multiple tubercles, my bad. Okay, you will have cavities or the holes, alright, in your lungs. Okay, so imagine you have a lot of cavities in your lungs. Your lungs is progressively destroyed. You can die from it. So this is the infectious disease in your syllabus 2 note. But there are treatments. So what are the 3 treatments that we have? Treatment for your TB. First one, I want you to take note. I'm going to erase this toucan. Enough. Need space. I want to talk about antibiotics first. Because bacteria, like Mycobacterium tuberculosis, can be treated with antibiotics. But I'm going to introduce more. Okay? Famous antibiotics that we learn in syllabus. is the penicillin as well as rifampicin. So I want to go through all this right now. First one in the syllabus to know is penicillin. And there is always the first phrase that we learn penicillin does, right? Penicillin actually prevents cell wall synthesis. So the mode is that, again, first primary mode is that they prevent cell wall. Okay. But the question is, what is always the phrase that will prevent the formation of this cell wall synthesis? First one, let's go through. For your this antibiotics, penicillin, what does it inhibit? Synthesis of, again, oh sorry, what is the enzyme it actually inhibit, Megan? Very good. So again, it inhibits transpeptidase enzyme. We learned under bacteria. Can you tell me, Myra, what does transpeptidase do? Synthesize, cross, links between numb and neck residues, right? But my next question is, do you know what is numb and do you know what is neck? Don't know. By the way, for people who do not know what numb and neck stands for, alright, numb actually stands for the N-acetylglucosamine. Okay. Sorry, NAG stands for N-acetylglucosamine. Okay. Neck actually stands for, NAG represents N-acetylglucosamine. Your NAM stands for N-acetyluramic acid. The name itself, uramic acid. Okay, but anyway, you always say inhibits crosslinks between numb and neg residues. Alright, so what does it actually do as well? Stimulates secretion of what? Hopefully you know what is autolysins. Here is the next row for your penicillin. Okay, so stimulates release of autolysins, and autolysins are the one that makes small pores on the existing cell wall. Idea is that this bacteria will be more prone to undergo this osmotic lysis, especially when you have increased turgor pressure. So 2. Rifampicin prevents protein synthesis when it enters bacterial cell. Same thing, you need to know what rifampicin do. It inhibits a particular enzyme. Any idea what is it, Valentia? Give me a hint. It prevents formation of RNA. So it inhibits what? Yes. In fact, it's a competitive inhibitor of RNA polymerase. So whenever we have here, we inhibit RNA polymerase, you inhibit this mRNA being synthesized. Sorry, RNA being synthesized. Okay? Okay. There are actually 2 more I want to introduce. Other effects, please take note, an antibiotic that we also... Oh crap. Oh my god, I never see the time. Oh my god. Okay, but never mind. Another antibiotic is the isoniazid. Isoniazid is also an antibiotic used to treat your TB. Alright, so do take note, this one actually prevents synthesis of cell wall components such as this glycolic acid. So idea is, you can see, this will definitely enable phagocytosis to occur, right? Give me a moment. Okay. So it enables formation of what we call phagolysosome. These are the 3 that I want you to take note. But something that is not in your notes is, how come this TB, right? Why do we always need to have a combination treatment of TB? That means we need to use more than one, okay, or more than a single antibiotic treatment. I do not have this. There are actually 5 reasons. You can write as your heading. I lazy, right? So the idea is that how come we need multiple, okay, or more than a single antibiotic treatment to reduce impact of antibiotic resistance. Okay? Repeat. Why more than single antibiotic treatment to reduce impact of antibiotic resistance? So you can see all these different antibiotics. I purposely give you 3, but they actually have different modes of action, right? Or So again, can you write different antibiotics act at different targets? I repeat, different antibiotics act at different targets, or you can say different modes of action. To increase chances of killing bacteria. So this is reason number one. To increase chances of to increase chances of killing bacteria. Second reason, number two, if mutation occurs, bacteria is unlikely resistant against all antibiotics. Even if mutation occurs, bacteria is unlikely to be resistant against all antibiotics. Okay? So again, number three, there will be, hopefully you know, because I have higher chance of increasing bacteria, your single antibiotic, right, is usually a very long treatment time. But if I use this, right, multiple antibiotics to treat, let's say, a single bacterial infection, there is actually a shorter treatment time. So the third one again, shorter treatment time. Since single antibiotic has longer treatment time and may not be effective in killing all bacteria, if resistance develops. Okay. Next one, another advantage of using multiple antibiotics is combination treatment helps eliminate bacteria more quickly. Since resistance to different antibiotics involves more genes to undergo mutation, and since resistance to different antibiotics involves more genes to undergo mutation, hence less chance of resistance occurring. Okay? So there's your antibiotic resistance. Again, in your syllabus, I just need you to know there's 3 types of antibiotics and how they work. Antibiotics will not be effective against viral infection. Why? You should know already, antibiotics target cellular components, alright? But viruses, again, one particular feature viruses have is what, Yunshin? Do they have cellular components? What do you call them? Acellular. So that's why there's nothing the antibiotics can target against. But for bacteria and your antibiotics, they're all targeted by vaccination. Vaccination, please take note, is a type of artificial, active immunity. When I say artificial, that means come from an external source. When I say active, that means you're going to involve T and B cells, but ultimately you are trying to create memory cells. So vaccination ultimately is trying to develop immunological memory, because you're trying to simulate that of an infected, or of an infection that the person has undergone, and then you're starting to develop all your these immune cells. So let's say we can have a faster secondary response if you are exposed to an actual, virus. So I say in syllabus, I'm not going to go through this because it's already in your summary slide that I already sent you. There are 3 types of vaccines you are expected to know in syllabus. 3 types are this live attenuated vaccine, your inactivated or killed vaccine that involves a killed pathogen, And third one is mRNA vaccine. Okay, so these are the 3 vaccines that we have. I want to talk about this. I already have, okay, the summary slide, the table that showcase the difference between these basic types of vaccine. I'm not going to go through. What I want to go through is this mRNA vaccine. mRNA vaccine, like you see your Pfizer and your Moderna, alright? Please take note. You notice how they will actually take an mRNA that encodes for an antigen or a glycoprotein of, let's say, a virus or a pathogen. Idea is, when we have here, okay, please take note, do you see we have this mRNA? mRNA, right, will actually must actually enter this cell, because only within the cell you have the ribosomes to translate and code for the antigen. So you express on the surface of a host cell that will simulate that of what? An infected cell. You are trying to pretend that this cell is now an infected cell. You understand? So that can stimulate your adaptive immune response, which will eventually form your memory cells. Hopefully you know memory cells is for what? Cheryl? Immunological memory. Give me a moment. Hello? Next one. So mRNA vaccine, right? You can see I have here, you must simulate that of an infected cell, so you can stimulate this antibody secretions and eventually memory cells. But however, there's an advantage and disadvantage, right? So let's learn now. There are 3 advantages of this mRNA vaccine. First advantage of mRNA vaccine, what do you think it is? Yunshin? Oh, my eyes here. 3 advantage, name it all. First one, it is, you think about this, every time they release that vaccine, Pfizer, Moderna are always the first ones, right? Because it's sorry, I can't. No,
long lasting.
No masking? How what?
Long lasting.
Oh no.
It's not long lasting.
So one thing we need to take note of, again, mRNA vaccine, right? One advantage is that it's, again, very fast to generate. Okay? So it's fast to generate. Number 2, simple to mass produce. Simple to mass produce. Number 3, safer, since there's no reversion to virulent form. You are not injecting a virus or a pathogen into the body. You are just injecting an mRNA. So it's safer since there's no reversion to virulent form. Okay? Next one, disadvantage. What's the disadvantage? Do you see, right? I cannot just inject, right? This mRNA must be uptaken. What's the chances of uptaking an mRNA? It's actually quite low. Okay? So, one disadvantage is it may not elicit a very strong immune response, since it does not contain antigenic components. So may not elicit a strong immune response, since it does not contain antigenic components. Number 2, do you agree you need to rely on the host cell machinery? Then you can express the antigenic components. Without the ribosomes or host cell, you cannot synthesize that one. So that's why, last one, need to rely on host cell to take up the mRNA and utilize host cell translation machinery to translate antigenic components. Okay? Yeah? Okay, so what is the advantage of having vaccines? I already have here, first of all, vaccines are long-lasting, but vaccines only target very specific cells. So one counter or pitfall that we have for vaccine is that, let's say if this virus is rapidly mutating, like HIV. Cheryl, why HIV rapidly mutates? Like undergo antigenic drift very rapidly. Why? You? Because no proofreading in? What enzyme? You must specify. Reverse transcriptase. Understand? So if you rapidly mutates, right, usually the vaccine is not going to have a very high efficacy. Alright? Because it only targets those that, again, for a particular type antigenic component. Agree? So same thing. The second thing this vaccine try to always undergo is always this. This is the number one thing. If you see this slide, make sure you take note of. It's very important. It's trying to create herd immunity. Idea of herd immunity is that the proportion of people that are immunized is so high such that it will also protect against, okay, those that are not undergoing vaccination at all. The idea is that you are trying to break transmission cycle. What is break transmission cycle? Break transmission cycle means the virus cannot find susceptible hosts sufficiently to infect and then increase its viral load to spread from one person to another. You understand? So I repeat, herd immunity is always the fundamental thing vaccination programs are trying to achieve, and that is to break the transmission cycle. These are always two key phrases that we mention for this vaccination. Okay? Now, so this is what I summarized. Now, because of time, I only can ask you to star. The answer sheet that I already attached is going to contain the things that I mentioned. But can you help me star this respective question so you can use this as a studying material? Question 2B, 2C, 2D. Please also help me star question 3A part 2, 3A part 2, 3A part 3 and B. Please also help me star question 4A, okay? Question 4A part 2 and part 3. You can use this as your studying material. So I gave you another thing, which is your MCQs. I feel like this chapter can be a bit wonky, so that's why I've sent out a list of MCQs for you to practice if you want to test on your content knowledge. Alright? Now, so take note. With that, I end off here today.